video, we're going to talk a little bit about the cell cycle and mitosis. So the cell cycle can be divided up into two major phases, interphase and M phase. Now both of these can be divided up further. So interphase consists of G1 phase, S phase, and G2, and M phase consists of all the phases of mitosis as well as cytokinesis, which is separate from that. So starting off with G1, or interphase in general, the cell is preparing to divide, while M phase is where the cell is actively dividing. Starting off with G1, which stands for gap 1, the cell is getting ready to actually replicate its chromosomes. So during this phase of the cell cycle, the cell is responsive to growth factors, and so something can tell the cell that you should actively divide during this phase. It's preparing to synthesize DNA, so you have DNA or proteins being synthesized that are going to be involved in the S phase of the cell cycle. S phase stands for synthesis. This is where the DNA is actively being replicated. So up here, the chromosomes don't have the sister chromatids present. After S phase, the sister chromatids are present. In G2 phase, the cell is, which stands for gap 2, the cell is preparing to actively divide, and then you go into M phase. So at the end of G2 phase, the cells, or G1 through G2 really, the cells are not condensed. We'll talk a little bit in a little bit what each of these phases of mitosis are doing, but just note right now that mitosis is talking about what the chromosomes are doing. Cytokinesis is saying what the cell is doing. So actually separating into two different cells is cytokinesis. All right, and then you notice that there's another phase over here known as G0. G0 represents a phase of the cell cycle where the cell is not actively dividing. So everything I've talked about before, the cell is either preparing to divide or actively dividing. G0 can be a phase that a cell goes into, and it may never leave G0 again. It's just never going to divide. Or it may enter G0, but some signal tells it to come out of G0, and you end up in the cell cycle again. All right, so this brings us to mitosis. So mitosis, remember, is the behavior of chromosomes. So mitosis is going to start over here on prophase. I'm just leaving interphase in here to kind of contrast it with the others. During interphase, the chromosomes are not condensed. You have centrosomes, but the centrosomes there's no spindles coming from them. They are not at the poles like you'll see during the active division. You'll see a nucleolus in the nucleus, and the nuclear envelope still intact. A few other things that's not in this. You have all the organelles being present. Some of the organelles are going to go away during prophase and then reform, or during mitosis and then reform after mitosis. So once you've reached the prophase, you are going to start to see the chromosomes start to condense. So you're going to clearly be able to see the two sister chromatids. The centrosomes are going to start to move away from each other towards the poles, and you have the mitotic spindles, which are going to connect to the chromosomes as well. The nucleus can still be intact at this point, but it's going to start dissolving such that during metaphase, the nuclear envelope is completely gone. These chromosomes are lined up in the middle of the cell at the metaphase or equatorial plate. The two centrosomes are at the two ends of the cell, and then the mitotic spindles are attached to the chromosomes at their centromeres. Now, once you get to anaphase, the sister chromatids separate. So this is important. It's not the homologous chromosomes that are separating, but the sister chromatids that are separating. And they're going to start moving towards the poles and away from each other. And then you go into telophase, which is essentially the reverse of prophase. So you have the nuclear envelope reforming. The chromosomes are starting to decondense. You don't have the spindle fibers present anymore. and the other organelles are starting to form. Now, sometime during this process, cytokinesis is going to begin. So cytokinesis can start during anaphase. Typically, you see it more during telophase, but it can last longer than the actual division. So cytokinesis is a separate process where the cells are dividing. All right, I hope this is helpful, and I will see you later.